But the point of doing these high MOIs is such that on average you'll have at least one. And so if one gets in and it protects from the other ones, who cares if there are more that are there? And so it's not going to change the absolute interactions between the two. Because the virus is just, it's diffusion. It's completely, it's a random interaction process. And so there's no will of the virus to get to the cell. And in most cases, the cells can't run away. There are a few exceptions to that. But it's always biology. There are always going to be exceptions. But for the most part, a pulsar distribution is a good way of describing these things. Any other questions about that? Yeah.
If you're actually doing something like this in the lab, you have to take into account like infectivity and how much of your virus is active when you're doing like a calculation to figure out how much virus you need.
Yeah, so the question is, I'm just repeating it, so it's here. So do you have to take into account, and again, I'll paraphrase, the infectivity of what you're looking for. So it's actually not the number of particles, it's the number of infectious particles. And so basically you will have had to have done some kind of infectivity assay before you do your one-step growth curve. And again, this is what people in my lab do a lot of, is to try and figure out how many you have to start with, and then using that to do your one-step growth curve. Okay. So there are a few more things I wanted to talk about review-wise. So we already talked about virus discovery. Basically, really small and causing disease. I think it's a really open question. And now, how the heck would you ask a clicker question or an exam question about this? Other than what other people thought. I don't think it's probably really appropriate. I think it's a really cool question to talk about, but I'm not sure we can cover that too much more detail. Cool things to do with viruses, yeah, there are a whole bunch of them. I just got started on some of them. Phage therapy, gene therapy, cancer therapy, etc. Basically, sort of take-home message there is viruses have a bad rap. Everybody always thinks about them as causing disease, but I like to think in a much more general sense. And then in terms of details, the plaque assays, again, that we talked about, and the one-step growth curves. Somebody come up to me in office hours and ask about, well, what about how you doing plaque assays and having your culture that's growing at the same time? How does that all work? So just the explanation that I gave there is that the one-step growth curve, kind of getting back to your question a little bit earlier, is you already know how much you have, because otherwise you couldn't be putting your MOI together, and you've gotten how many virus particles you have usually by some kind of plaque assay. So you've got numbers that way. And then you do a mixture in liquid culture at your high MOI, and then follow what's going on in that liquid culture by taking out samples on a particular time basis and doing infectivity assays or DNA assays, whatever else you're looking for, over that period of time. And if you're doing plaque assays, getting the results of those plaque assays is going to take way longer than your growth curve does. Because the plaque assays, you've got to go through multiple rounds of replication. In a one-step growth curve, you should have one round of replication that you're talking about. Does that make some more sense? So the plaque assays and the one-step growth curve are really very separate from each other. Plaque assays are counting. One-step growth curve is a synchronous infection that you can then follow and get some time off of. Okay, that makes some more sense. It's a bit of a complicated concept for me and probably for other people as well. So today we'll talk a little bit about replication strategies, and this gets back a little bit to your question about interactions between viruses and hosts. We'll talk a lot more about that as we move along. Most of the time we're going to talk about virus structure today. A little bit about how virus structures have been determined, mostly having to do with electron microscopy and X-ray crystallography. Icosahedral symmetry is an absolutely wonderful process that, at least for me, is hard to follow, which is why I bring lots of three-dimensional models here, and I've got a whole bunch more in my office. For the problem set, I used to have people build icosahedra and bring them to me. I now have a whole collection of icosahedras. I've stopped doing that, but it's a really great exercise to go through to follow icosahedral symmetries, actually put these things together. Key numbers, which are a really nice way of describing icosahedral symmetry. We'll look at a number of examples, and then a lot less is known about the envelope viruses. These are really kind of squishy and mushy for the most part. Cytomegalovirus is a really nice example of that. And then how everything gets put together. It's a really unique process in terms of viruses, because the capsid, like we talked about I guess on Monday, we talked about capsid encoding organisms as being the viruses. How do you put that capsid together? How do you make the bag around the nucleic acid? This is a purely viral process of making that virion. And then we may or may not get around to talking about virus taxonomy. If we don't, it's not such a big deal because virus taxonomy is a mess. So key concepts in terms of the overall classification of viruses really has to do with the messenger RNA. And all viruses that we know of today require cellular translation. So somehow they've got to make a messenger RNA. And so David Baltimore came up with this classification in the 1970s called the Baltimore classes, which have been really very, very useful in terms of thinking about viruses. So we'll talk about those a bit more later on. In terms of virus structure, it's basically about simplicity. Icosahedra are simple, helices are simple. And then the taxonomy, some of the structure, some of the genomes. So what about virus replication? This is the overview for basically all viruses. I showed you that one circle when you think about a virus. Again, it's not just the virion, it's the whole process. That whole process consists of first binding to the outside of the cell. This is the virion binding the outside of the cell. And this is that random process. This is what's described really well by that Poisson distribution. It's the interaction with something on the outside of the cell. That something can basically be pretty much anything that's on the outside of the cell. Proteins, lipids, carbohydrates, you name it. And so the evolutionary aspects of these viruses and how these viruses are interacting with cells have basically run the whole gamut. Everything that's on the outside of a cell could be something that a virus interacts with. How these viruses interact, we'll talk about the individual viruses, but just in general, what cells viruses bind to is called tropism. And so if you have a particular kind of virus, we'll take an influenza virus, what kinds of cells does it interact with? Epithelial cells and airways. And so that would be the tropism of influenza viruses. And that's because of the particular cell surface molecules the virus interacts with. Once it interacts, then you've got to get the viral genome inside the cell. This happens in a number of different ways. Some of my favorites are the drilled process, which is what happens with some of the bacteriophage. Bacteriophage T4 is a really nice example of that. You've got a really tough outside of the cell you need to get through. But lots of other different ways, actually damage to the cell, which happens with a lot of plant viruses. Fusion, this is really easy if you're an enveloped virus, because your enveloped virus is a membrane, and membranes love to fuse with each other. So once you have one membrane come down, fuses with the next membrane. And in some cases, actually, that capsid has to be degraded in order for the genome to get inside the cell. So you remember that it's all about getting the genome inside the cell. You don't really care about the capsid, and this is what Hershey and Chase showed when they were showing that DNA was genetic material. Once you have your genome released inside the cell, then you make these so-called early genes, and this is coming back to that one-step growth curve. These are the things which are made first. And these are almost always genes which are going to be important for replication of the virus genome. If you think about early gene expression, those are going to be the polymerases, which are important for replication of the virus genome. So early genes going to be important for replication. Once you've made more of the genome, you've got to put a bag around that genome, and that's the late genes, which are here. These late genes are usually the ones which are important for putting together your capsid, putting together the virion. So early make genome. Once you've made the genome, you make the bag to go around it. And then once you've made these proteins, they need to be put together, and then somehow they have to get outside of the cell. And that somehow getting outside of the cell could be sort of what classically people think about in terms of virus replication is lysis. So make all these particles inside the cell blasts open, the viruses all get released. That's what happens particularly in the case of some of the naked viruses, those that don't have envelopes around the outside. And just by looking at one-step growth curves, if you look at intracellular virus, you lyse a cell separately, you can see a difference in terms of the timing for a lot of these. Budding, this is what happens with most enveloped viruses because they have an envelope. That envelope is something which gets picked up from the cell. Very often it's the plasma membrane. So it buds off just like vesicles are budding off in, say, neurotransmitters and these vesicles, etc. Sometimes you actually have viruses going directly from cell to cell. And in many cases, we have what are called viral vectors. So they basically will take virus from one cell and move it to another cell. They'll take virus from one organism and move it to another organism. We'll talk a lot more about vectors later on. So that's the overall process. We'll go through each one of these things every time when we come up with a new and different virus family here. What are those different virus families? Well, again here, it's all about the messenger RNA and how this actually gets made. So we mentioned the early proteins. This is what gets made as soon as the genome gets inside the cell. Well, what is that genome and how do you make those early proteins? There are quite a few viruses, and in fact probably most of the viruses in plants and a lot of the viruses that infect animals, which have a single-stranded RNA, which is what's in the virus particle. And that single-stranded RNA can code directly for proteins. And if anything, this is the most efficient way to make viral proteins, because what comes into the cell after it comes out of the capsid can be translated immediately. So it's great. It makes your proteins, those are the early proteins, which again, usually you need to replicate your genome, etc. So that's one way. But it turns out there are lots of other kinds of viruses as well. We also have viruses that have double-stranded DNA inside their capsids. Those double-stranded DNA viruses then will get transcribed, the messenger RNA, and this is the process extremely similar to what goes on with cellular replication in terms of making proteins. There are a few cases where you've got partially single-stranded and partially double-stranded DNA. We're not really going to talk about these very much. George Kassoon will talk about some of these really bizarre viruses, which are single-stranded DNA, which gets packaged inside the virion. Why you should have single-stranded DNA rather than double-stranded DNA, because it absolutely has to go through a double-stranded DNA intermediate. I've got some theories about why this is, but they're currently just theories. Then you also have a few and infamous kinds of RNA viruses, which are the negative strand RNA viruses. Influenza, Ebola, a lot of the nasty viruses are what's called negative strand RNA viruses. Why are they called negative strand RNA viruses? That's because this RNA doesn't code for protein. It codes for the complementary strand, which is going to be encoding your proteins. So this may seem a really inefficient way of bringing your genome inside the cell, but as we'll see later on when we talk about some of these viruses, it turns out to be really useful for some aspects of virus replication. But because this RNA doesn't encode proteins directly, it can't go to the ribosome and make the early proteins. All of these viruses have to have a viral RNA-dependent RNA polymerase that comes with the genome inside the cell when it gets released from the capsid. So here's not just the genome that comes in, there has to be a protein. Because if there's not a protein, you're never going to make the positive strand is what you need to make your proteins. Turns out the same thing is true for double-stranded RNA viruses, even though In theory, one of these is a positive strand that could be translated directly. Turns out that you also need a viral RNA-dependent RNA polymerase. Those of you who've been watching, notice there's one down here at the bottom. And this is basically why Baltimore came up with this in the first place. And that was the so-called retroviruses. These are viruses that have a positive strand RNA that is in the virion. That's what gets released inside the cell, but strangely enough, it doesn't get translated. It gets made into first single-stranded and then double-stranded DNA before the messenger RNA is made. And that process of going from RNA to DNA also requires a viral protein to come in with it, the reverse transcriptase. And so these are the virus types which have always a viral protein which comes with them. Yeah?
So the viral RNA-dependent RNA polymerase or the reverse transcriptase are packaged inside the capsid, and they're injected into the cell when the virion infects the host cell?
Yeah, so the question is, do these proteins get injected with the genome at the same time? And the answer is absolutely yes. You can't have replication of these viruses if there isn't another viral protein that comes with them. So a lot of times when people think about viruses, it's just the genome that comes in. Well, in this case, it's the genome plus these extra proteins that it happens to be. And that also tells you a little bit about how the genome is going to be getting into the cell in the first place. If you have some kind of injection process like you have with some of these bacteriophage, there's only enough space for a DNA to come in. And so you couldn't be injecting a protein at the same time. So there are a number of different ways of looking at this. This is just the image from the Flint textbook. Again, it's all about the messenger RNA. I don't expect you to remember all of these different classes. I can't remember them myself, but that there are 6 of them, I think, is a reasonable thing to think about. So we've got viruses which have positive strand RNA, viruses which have negative strand RNA, double-stranded RNA, double-stranded DNA, single-stranded DNA, and RNA that goes through this reverse transcription process in order to make the messenger RNA. One of the things about this slide, which I think is a little better than the one from our textbook, is it also shows that for these positive strand RNA viruses, it's really easy to make protein, but it's actually a lot harder to make the positive strand RNA that you need as your genome for the next round. So you always have to make a negative strand RNA as well. So it's not just, you can't get away with just having that positive strand RNA, except with the one genome that comes in, and then that one genome goes back out. And that's not the way that these viruses replicate. This is a diagram that I put together. Again, just one more for your notes. Here's the messenger RNA right here. The reason I like this one is because I put my capsids around all of them. So little blue things are all the capsids. And just that lets you know what is being packaged here. Everything which is black is your DNA. Everything in red is your RNA. Any questions about this process action?
What was the you said there was maybe a 7th one now?
I didn't say there was maybe a 7th one. Some people call them 7th ones. So if we go back 2 slides, there's this one right here. Partially single-stranded, partially double-stranded DNA. Some people will call this a 7th class. I don't like it very much, and it turns out that it's really a subset, these single and double-stranded DNAs, of these retrovirus-like processes. So, yeah, some people talk about 7 classes. In this class, I'll talk about 6. And Baltimore only came up with 6 originally. And so it was later that 7th class came in. And 5A, partly because people can't agree. They'll call it 5A, they'll call it 7, you name it. So it's kind of a mess. So I like to stick with old school 6. Works for me. And that's also why we've got just 6 on this slide as well. Okay, questions about that? That's basically chapter 1 of the textbook. Took us all week to get through. We'll take a little shorter time for some of the other chapters we move through. But I think it's important because these are kind of general aspects that we'll be talking about for all the different viruses that we move through here. Any more questions about this? Hopefully I'm kind of beating the dead horse. Good. So talk a little bit about virus structure. Again, the idea here is really simple, as simple as possible. And this gets back to the fact that most viruses have relatively small genomes, and DNA is really, for that matter, RNA is really kind of inefficient. You need 3 nucleotides to encode 1 amino acid. And all of these capsids, they could be nucleic capsid just binding to the nucleic acid, or you have a nice helical or icosahedral structure around the outside. You want to have small numbers of proteins, and preferably exactly the same protein or very similar proteins to each other. So that's the first aspect of virus structure. The second one is a really interesting aspect about viruses, and what people call the virus capsid paradox. Because on the outside of the cell, you want the capsid to be really stable and be able to protect the genome, which is inside, the nucleic acid. However, once you get inside the cell, you want to be able to release the genome. So there's this whole interesting balance, and again, this doesn't happen evolutionarily, such that on the outside of the cell, the virus is really, the virion, I should say, is really stable. On the inside of the cell, the virion falls apart really easily. And so this is one of the aspects about virus, and particularly virus structure, that we don't particularly understand very well. And then we'll talk about icosahedral symmetry and quasiequivalence. How do we know about these structures? Basically, with these really cool, really expensive instruments. This one's actually downstairs in Science Building 1, just over here on the other side of campus. FBI Techni F20, it's about 10 years old now. This is the latest, greatest electron microscope made by the FEI company, which is based in Hillsborough, just down the road. And they have one of these that's currently installed up the hill at OHSU. It's going to be in CLSB in the relatively near future. Very high resolution microscopes, really critical for looking at individual virions. What do these virions look like? We looked at some of these already. Long filamentous particles, icosahedral particles, some of them connected to helices as well. Some of these are your classic enveloped virus particle, but they also have proteins bound to the nucleic acids on the inside. This is a nice example of a pox virus, and here's a naked virus particle. The way that you visualize these things is you're not actually visualizing the virus particle itself. You're visualizing the negative stain, which you put on the outside. These are usually heavy metals, which are going to absorb electrons. Our favorite is uranyl acetate. Using uranium, you can also use tungstate. And so if you come by the Geiger counter with our lab and look really, really carefully, you can find some of this non-enriched uranium, and we're not in the nuclear talks with Iran at all. So, but yes, so basically you're looking at the outline of your virus particle in this case. You can also do what's called positive staining. It's a heck of a lot harder to do. We do negative staining all the time because it's easy. Positive staining is a lot more challenging. Usually you have to get your cell or your virus prep, cut it with a very, very, very fine knife. Usually these are tens or hundreds of nanometers. So 10 to the minus 9 meters that you use for making these thin slices. Then stain again with some kind of heavy metal that's going to absorb in the electron microscope. And here's an example of some HIV particles associated with a cell. This is a classic HIV nucleic capsid on the inside with its envelope around the outside. So these are great for just identifying individual particles. It doesn't tell you anything about the high resolution structure. And to get these higher resolution structures, basically you need one of those Titan Cryos microscopes, which, oh, by the way, the size of these things is about 5 feet higher than the classroom here. These are really big instruments. And you can use what's called cryo-electron microscopy. And in this case, you take your virion preparation and freeze it with the cryo bio is all about, and then image lots and lots of these particles. And we'll look at how that happens in just a second. Probably the most time-consuming way, but the highest resolution way of looking at things. Let's say highest resolution is the difference between something being there and something not being there, is X-ray crystallography, which works extremely well for very, very few characterized viruses. I think there are probably about 10 total virions which have been crystallized and then their structures solved by X-ray crystallography. But the main way that people use to get high-resolution structures of individual virions is cryo-electron microscopy, and usually what's called single-particle reconstruction. So you take your virus preparation and you freeze it in vitreous ice. And so this is really important. You do a very, very rapid freezing to get this. You don't want ice crystals. If you have ice crystals, they're going to break apart your virions. You do this in the case where you don't get ice crystals. The way you can do that is by freezing really, really fast. And really fast, I mean on the order of thousands of degrees a second cooling that you do for these things. And there are special instruments that do this. So you've got your virus particles in vitreous ice, a whole mixture of different virus particles. And then basically, you take each of these individual virions and separate them all relative to each other, then try and line them up in the same way. This is all done computationally. You see this guy's got these two things sticking out down here, two things sticking out down here, et cetera, et cetera. And then, in the computer, average all of these things together. And in that process, you end up with a three-dimensional reconstruction of each of these particles that you started with here in the first place. And again, this is stuff which is being done up the hill at OHSU, soon to be done down the hill at the CLSB. And basically the way most of these structures we'll be looking at have been solved. You can also do X-ray crystallography. This depends on having a crystal. And that's really the major problem with X-ray crystallography, and particularly with viruses. Yes, some viruses you can crystallize. TMV is really the exception to that. That was why they said it wasn't alive. You can't crystallize life. But for the most part, it's really hard to crystallize any of the whole virions. Very often, however, what people will do is crystallize the individual proteins that are making up the capsid, and then get high-resolution structures for those, and then use something like the cryo-electron microscopy to put it together in order to figure out what's going on there. But if you do have a crystal which you can form, you can do diffraction, collect the spots, do a lot of computation, then you get an electron density map, which is what's shown here in the blue on the outside. That's the real data you get from X-ray crystallography is this electron density. And then what you try and do is fit your amino acids into that electron density. And hopefully you've got electron density that have a little blob right here. You say, oh, that looks like it should be a isoleucine, for instance. And then you trace your way along the whole process. Here's a big blob that looks like it would be a phenylalanine. And so you know the sequence of the proteins that you're looking for. You try and fit them to the data. Once you've finally done this, then you strip away all of the electron density and say, wow, isn't that cool? Look where we've got our alpha helices and beta sheets as well. This is just the model which best fits the data. So what do these things then look like? Smaller is better. So again, this whole keep it simple, stupid process. A number of really small viruses, particularly some of the single-stranded DNA viruses we'll talk about, some of the small RNA viruses we'll talk about a little later on, can actually self-assemble. And this gets back to the polio example that we talked about last time. You can purify the proteins, you can purify the nucleic acid, you can put them together, and you get infectious virion, which is really pretty amazing that this process, in fact, works. This is true for very few of the viruses which are out there, and usually the extremely simple ones. I mentioned already nucleic acid is just too darn big. You need 3 nucleotides, which No. How many nucleotides per turn in E-form DNA? 10 nucleotides per turn. How many angstroms is that? Yeah, about 34. The smallest virions are about 34 nanometers across. So you'd have 100 turns, and that's it. Well, 100 turns, 10 bases each, that's 1,000 nucleotides. It's going to be really tough to fit much into that. That would be 300 amino acids, okay? Well, you need to be making your replicase proteins, et cetera, et cetera. So turns out that making the capsid protein is often a huge proportion of the genome of the virus. In some cases, here it's got a third. In some cases, it can almost as much as half of the genome of your virus is made up of this capsid protein. So how do you use as small amount of the genetic material as possible and still be able to make something which is going to protect your genome on the outside? And basically has to do with using that same sequence again and again and again and again and again. So how does that work? Basically, the way that they show it here is all of these little commas. So each of these little commas here is going to represent one protein and how it interacts with all of the other proteins around it. What's really nice about this kind of arrangement, here, sorry, where'd the pointer go? Hiding over here, here it is. This particular subunit here is going to interact with all of the subunits around it, just like this subunit here is going to interact with all of the subunits around it, which interacts exactly the same as all the other ones. You can have exactly the same protein, which interacts in the same way with all of the other proteins which are around it. And if you bind here, put A associating with B, just wrap this up, you end up with a nice helix. Helix is then a completely symmetrical interaction. All of these subunits are interacting with itself in the same way, except, of course, for the ones that are present on the ends. And so this is why you can get really great what we call genetic economy. One protein interacting with all of its friends in exactly the same way in order to get you a capsid. So this works okay, except, again, we've got this kind of end problem. So the other solution, which evolution has come up with over probably billions of years, is this icosahedral way of putting together particles. And just a reminder from geometry, way back when, my 5th grade daughter is taking geometry now. So when we're talking about an icosahedron, it's a 20-sided regular structure where you've just got 20 triangles all the way around the outside. And you can describe these icosahedral particles in basically 3 different ways, because there are 3 different axes of symmetry that you have. There's what's called a twofold axis of symmetry here. And basically what that means is if you take your icosahedral symmetric object, let's take this one right here, and flip it 180 degrees, you end up with the same thing. So that's your twofold axis of symmetry. You also, with these guys, have threefold axes of symmetry, which is you now rotate it 120 degrees, you get exactly the same thing back. Or you also have a 5-fold axis of symmetry, and there you just can rotate 5 times, you end up with exactly the same thing back again. So any icosahedra is going to be defined by these 3 axes: 2-fold axis of symmetry, 3-fold axis of symmetry, and a 5-fold axis of symmetry. We'll talk a lot about these different axes as we move on through here. Why icosahedral symmetry? Well, if you think about the other regular 3-dimensional shapes that you can get, tetrahedron and cube, they're just too small. You can't fit your nucleic acid, your inefficient nucleic acid inside. So icosahedra work really well for viruses. And now if you take these little commas that we had before, that we're using for our helical symmetry, and now arrange them with each other. Here, this guy interacting with this guy across a 2-fold axis in exactly the same way. Here's this guy interacting with this guy across a 2-fold axis in exactly the same way. All 3 of these guys are interacting with each other around a threefold axis in the same way. These three guys interact with each other across a threefold axis in the same way. And at the fivefold axis, you've got 5 of these guys that all their tails are interacting with each other. But each of the fivefold axes are interacting with each other in exactly the same way. So if you've got one protein that's interacting with 4 of its friends at every one of the fivefold axes of symmetry, you've got 12 fivefold axes of symmetry. 12 times 5 is what? 60. So the minimum number of proteins that you'll have to make an icosahedrally symmetric particle are going to be 60. You got a question? Yeah.
On the last slide, you had 6-fold
Yeah.
symmetry on there. Is that?
So the 6-fold symmetry is just basically a reduction of your 2- and 3-fold symmetry.
Okay.
So you can use that. The 6-fold really, almost no one's going to talk about that.
Okay.
Yeah. So in the simplest case, you've got 60 capsid subunits. Now, one important thing that I need to mention here is that icosahedral symmetry, which is 5-fold axis, 2-fold axis, and 3-fold axis, ignore the 6-fold, is something that can describe lots of different things, some of which are going to be regular icosahedrons, some of which aren't. And so this particular case, where I've got my soccer ball here, this has icosahedral symmetry, but it's clearly not an icosahedron, it's a ball. And so if you have a 3-fold, a 5-fold, and a 2-fold, that's going to be icosahedrally symmetric. An icosahedron is going to have triangular faces all the way around the outside. And there's a really nice description here at the Viper DB that I talked about before in terms of looking at these various different icosahedra. So why do we care about icosahedra? Well, because icosahedra and things with icosahedral symmetry are using relatively few number of subunits. Remember, it's 60 subunits, but they're all exactly the same. And you can make a relatively large volume in this process. So one of the nicest examples of these are some of the plant viruses, which have purely icosahedrally symmetric. They have 60 subunits. All of them arranged exactly that way that I was talking about. We know in this particular case for Cowpea Mosaic virus, it's one of the very few that we actually have a X-ray crystal structure for. This is the 5-fold axis of symmetry of this particularly icosahedrally symmetric virion. And you can also nicely see these 5-pointed stars, which are at each of those 5-fold axes of symmetry. This is great, works wonderfully if you have a really small genome. And that's true for some of these viruses. However, as soon as you start to get a larger genome, it can't fit into something that just has 60 identical subunits. So this brings up that question you had about the 6-fold axis a little bit earlier on, is this concept called quasiequivalence. And this is a mathematical concept that was brought up by Casper and Klug in the 1950s. And basically, it's a way of taking a icosahedron and basically making it bigger by having the same subunits, but interacting with each other in slightly different ways. So that slight difference here is now a 6-fold interaction. So you remember each of our little commas here that are normally interacting here with each other, the tails, 5 of them. You now slightly tweak the structure and now get them so you got 6 of them interacting with each other. They're similar, but not quite the same. That's where quasiequivalence comes from. Now you can basically expand these icosahedra as big as you want to. And that brings me back to the soccer ball, because in this case, we actually have 6 that come together right here. If you think about a soccer ball, it's got a lot of pentagonal panels and hexagonal panels, but basically put together by more or less the same process. So how do you think about this in a mathematical sense? Some of you may have noticed this T number up here at the top. This just comes from the mathematical relationship between the individual particles, I should say now the particles that are set up by these different subunits, and how those subunits are arranged on each of these different particles. So in this particular case, it's just a regular quadratic equation, h squared plus h plus k squared. All you need to do is get from one 5-fold axis of symmetry to the next 5-fold axis of symmetry. And depending on how many 6-fold axes of symmetry, hexagons you have between each of your pentagons, that's going to give you a description of what your icosahedral particle looks like. And again, going and making some of these things is an extremely useful way of thinking about this. But soccer ball is this example right here. Here we've got a 5-fold axis of symmetry up here at the top. Here is a 6-fold axis of symmetry. Here's a 5-fold axis of symmetry. To get from this 5-fold axis of symmetry to that 5-fold axis of symmetry, you've got to go via this 6-fold axis. So you make one jump, and that's the h direction. If you kept going here, you wouldn't get to the next 5-fold axis. You've got to go in a different dimension, in a different direction. That's going to be k. So now we move from here, one step, we get to the 5-fold axis of symmetry. That's an h of 1, a k of 1. 1 times 1 plus 1 times 1 plus 1 times 1 is 3. So this is a classic T equals 3 icosahedrally symmetric particle, and a soccer ball is a really nice example of that. It's not the only way that you can make icosahedra. There are lots and lots of different ways of making icosahedra with adding this quasi-symmetry. This example up here in the upper right is a T equals 4. Here we have it'll be easier here, 5-fold axis of symmetry here, 5-fold axis of symmetry there. We make one step. We don't have to change direction, so we keep going in the same direction. 2 steps. That's an h of 2, k of 0. So 2 squared is 4. It's a t of 4. And there are only certain allowed t numbers that you can have in terms of putting all this stuff together. Yeah?
So when you're counting this, you're only going from 5-fold to 5-fold.
Exactly. And so here's a nice example of that, a real example. This is Cowpea chlorotic Mosaic virus going from 5-fold axis to 5-fold axis. It'll be a little harder to tell here. This is a hexagon, and these are pentagons. So pentagon to hexagon to pentagon. So go 1, change direction, go 1. Same thing is true where you go this way. Go 1, change direction, go 1. These are symmetrical particles. So everybody got this down? How do you get to 31 from this? You don't. So,
Almost
sure.
right. So is the h the number of steps that you take kind of, and the k is like the times you change direction?
No. So h is 1 dimension and k is 1 dimension.
Oh.
And so when you change directions, you've gone from h to k.
Oh, okay.
There are 2 axes that you can think about. And if you lay this whole process out on a flat sheet of paper, that's exactly what you would see. Okay, so now, one of my favorite viruses, because I actually discovered it, and it was the first ever virus to have this kind of icosahedral symmetry. This is a T equals 31 symmetry. So how do we get to T equals 31? Well, you can try and do it from here. The 5-fold axes of symmetry are really easy to find. Look, you've got these beautiful projections around the outside here. But the question is really, what about the hexagons which are present in between? So it's a lot easier to look at it this way. And if I were to give you a question like this in an exam, I would give you one of these. So you need to get from one of these 5-fold axes of symmetry to another 5-fold axis of symmetry. So now, in fact, I'm going to go and use where's the pen here, because it's a little bit easier to follow. So we'll do a pen, and go from this 5-fold axis, we go 1, 2, 3, 4, 5. Now we need to change direction. So it was 5H. and 1K. So 5 squared is 25, plus 5 times 1 is 30, plus 1 squared is 31. So, and the largest, well not quite the largest, Pandora is a little bit bigger, but the Mimiviruses have massive numbers of these subunits all put together. They still have exactly the regular Icosahedral symmetry. You can define exactly where these come from. There are a number of variations on this theme. Anybody recognize what this is down here at the bottom?
Buckminster Fullers.
Pardon?
Buckminster Fullers.
The Buckminster colorings. This is from the Expo Center in Montreal. And sometimes I ask people to actually try and find the T number for this. And this is completely Icosahedral symmetry. Here's a Buckminster fullerene, nice T equals 3 particle. In many cases, there'll be a normal icosahedra that will have some extra bits of capsid proteins, which come together in the middle here. This is the case of 529. This is HIV1 over here. We've basically got an icosahedral shell at one end, a different kind of icosahedral symmetry at the other end. How this gets put together is a really interesting question. And then our lemon-shaped viruses that we work with in our lab might actually have some similar kinds of structures to this as well. And then, of course, our friend the soccer ball as being one of these. So let's look at a few more examples of these kinds of icosahedral virions. Again, classic kind of structure here. Polio is a really nice example of this. It's a single-stranded RNA virus. Here's a 5-fold axis of symmetry. Here's a 5-fold axis of symmetry. Here's one of those 6-fold axis of symmetry. So this would be what kind of particle? So T equals 1, 1, T equals 3 particle. Only polio is a little different because instead of having just one capsid protein that you put together for this, it actually has 3 different capsid proteins that go together for this. They're very similar to each other, and that's what these structures are that are shown down here. BP1, BP2, and BP3, they all have the same core structure, which fits together to put together your icosahedron, but then they've got these little extra things on the outside. And so this is what's called a pseudo T equals 3 structure because it has different proteins that are going together, but they're still assembled in exactly the same way as you would have if you just had one particular capsid protein. And one of the other things that I forgot to mention that's really cool about T numbers is if you know what the T number is, you actually know how many capsid proteins are put together into your shell. So T equals 1 had how many? 60, because it's just every 5-fold axis has 5 proteins associated with it. T equals 3, 3 times 60. It's 180. T equals 31. Anybody really quick? I can't remember off the top of my head either. But 31 times 60 is exactly the number of capsid proteins that you have on the STIV, the virus that I found in Yellowstone. So really straightforward way to get the actual number of capsid proteins that you have together there. So this was great, this was wonderful, and then people did an X-ray crystal structure of the SV40 virus. And it only has 360 protein subunits that fit into the electron density that makes the shell. Okay, so 360 subunits, your T number should be what? 6. How do you make 6 out of h squared plus h plus k squared? You can't. So it turns out that SV40 and a few viruses, this is much more of the exception, take the whole Quasiequivalence thing and really kind of throw it out the window, or what they do is they take these places we should have 6-fold symmetry for Quasiequivalence, because it'll be the same protein that's doing this, and they actually have pentameric proteins that go there. This is all one protein. It's just one protein that makes up the shell, the capsid in SV40. it is completely five-fold symmetric at these five-fold axes of symmetry. But where you should have 6, you only have 5 in each of these areas between them. And so what happens is you have these pentameric subunits that go where you should have hexameric subunits in the first place. And what this means is you've got one protein that actually has 6 different structures. Now, if you're back in principles of biology, say you have one amino acid sequence gives you one structure. Well, this is one amino acid sequence that's giving you 6 different structures. But all fit together, make this capsid structure. And what's interesting, it turns out, is that the structure in the middle of this protein has this beta-stranded structure that hopefully should look really similar to the beta-stranded structure that you have here in polio. Turns out that that beta-stranded structure is similar in many, many, many different viruses. So here's this really bizarre example of how you can have icosahedral symmetry. A much more straightforward example of how you have icosahedral symmetry is the adenovirus. This is type 2 adenovirus. This is a pretty big, relatively big at least at the time, non-enveloped virus or a naked virus. Here the question is, how do you get from one five-fold axis to the next five-fold axis? You see here are these what are called pentons, should be kind of a giveaway. And the fibers, you think about some of the classic pictures of viruses, they have these fibers extending from the five-fold axis of symmetry. So you need to get from a five-fold axis, and here's a five-fold axis of symmetry, to the next five-fold axis of symmetry. How do you do it? You go 1, 2, 3, 4, 5 in a straight line. That gives you what kind of T number? T equals 25. Now I tell you that the proteins that are present in the hexons, these hexameric units, are different than the ones that you have in the pentameric subunits. What does that mean? It will be a pseudo 2 equals 10 to 5, because they're different proteins which are being used, but assembled in a purely icosahedral fashion. To make this relatively complicated structure, you need something called scaffolding proteins that I mentioned, actually didn't mention, but they were on the slide earlier on. Scaffolding proteins are structural proteins, things that you need to make your virion with. They're late proteins, but they're not present in the final virion structure. And so you do seem to need to have these proteins to build your virion on, but then you lose them in the final assembly process. And probably the most spectacular example of processing that happens with these virus structures is first shown in the bacteriophage HK97, but has now been shown to be present in lots of different, mostly bacterial viruses. And these are structures. This is a pretty regular T equals 7 icosahedrally symmetric particle. Here's a pentamer. There's a pentamer. You go 1, 2, go across, get 1. But the more impressive thing about this structure, which also was solved by X-ray crystallography, is it turns out that the individual protein subunits that are making up this shell are covalently cross-linked to each other. And they're covalently cross-linked to each other in order to form this sort of chainmail structure in the actual virus particle. And nobody believed this for a long time. It took the graduate students working on this about 3 years to actually get it published. But what happens here, at the threefold axis of symmetry, you have a peptide cross-link that forms between the different subunits here. So this light blue cross-links to light blue, the purple cross-links to purple, and the green cross-links to purple, crossing over each other at each of these intersection points, either at the threefold axis of symmetry or wherever you have these structures coming together. What that means is you have this incredibly stable capsid structure, and it can be really, really thin. And so it's a really amazing process. And it turns out that just by bringing these side chains So they're, this is not the peptide bonds, but it's peptide bonds that form between side chains with each other. So acidic side chains and basic side chains, they get right next to each other and then form these peptide bonds. Already talked about some of the big viruses having basically identical structures to what we had before. This one has a pseudo P equals 169, so T pseudo 169, and then the Mimivirus actually has a T number of 1,179, but those capsid proteins are fitting together in exactly the same way for all of these things. To finish up, 2 minutes left. Helical symmetry. We already talked about this in terms of each of the individual subunits. We didn't talk about any examples. The Mosaic virus is probably the best understood of these. It does crystallize quite nicely. Each of these subunits is then related to all the subunits around it in an exactly equivalent way. The big difference is here at the ends. What do you do about ends? And so helical symmetry is really straightforward. It can extend basically as long as you want it to, but you've got to do something about the ends. And any time we've talked about this in molecular biology, ends are a real problem for your nucleic acid because exonucleases just love to chew in at ends of things. So we'll stop with this example of helix and icosahedra put together. This was a structure that was solved a year and a half ago where we have both symmetries together. Very true in bacteriophages, less so in animal viruses.